Welcome everybody to another KSVDL webinar. My name's Greg Hanselcheck and I'll be the moderator for today. Thank you to all our repeat webinar attendees. Thanks for taking time to be with us again, and for everybody or anybody joining us for the first time, thank you also for taking time out of your day to be with us today. A little bit of information for those first-time listeners is number one, if you stay with us the entire time, you earn 1 hour of continuing education. We will send you a certificate via email. Give us 2 to 3 weeks to get that to you, but you will receive one. If you haven't received one in about 3 weeks, please contact me and we'll get one sent to you. These are also recorded, and one of the recordings will be on the KSU CVM Continuing Education website, and it'll be with a small, short quiz, 4 to 5 questions. If somebody watches the video there and then answers the short quiz, again, they'll earn 1 hour of CE. This video will also be planted on KSVDL YouTube channel in a couple weeks. If somebody watches it on the YouTube channel, there isn't any continuing education that actually goes with that. So just a little heads up on that. The way we plan on these webinars is to have the presentation, and then we try to leave a little bit of time at the end for questions. If you have questions during the presentation, please enter those either in the chat or the Q&A at the top of your screen there. The difference between those two is the Q&A is going to be more anonymous, so if you don't want somebody or everybody to know who's asking the question, that's a good way to send in your questions. If we run out of time or we don't have time to go over all the questions, at the end of the presentation will be contact information, email for our speaker today. Please feel free to contact him with any questions that you might have. The last thing is you'll also be asked to complete a survey at the end of this, and please fill it out. It's very short, and there's a couple things we ask is, number one, what are we doing right for these webinars? What could we improve on? And then really the important question is, give us some ideas on some topics that you would like to hear a webinar on in the future. And again, we're not going to be able to give advice on treatment or those kind of things. It's primarily diseases or surveillance, anything like that, and then a little bit of diagnostics. So any ideas, we really appreciate any input from you all. So with that, we'll start with today's presentation. It is my pleasure to introduce Shane Hesting. Shane's a longtime collaborator with the KSVDL. He's actually the Wildlife Disease Program Coordinator for the Kansas Department of Wildlife and Parks. Shane has a bachelor's and a master's degree in biology and an emphasis in environmental biology. And I've got to read this. His thesis for his master's was environmental correlates of cove fish assemblage structure in Melbourne Lake. So we won't go into that today, but someday I'm going to have to ask Shane just exactly what the heck is that. So kind of interesting. He has about 16 years of experience conducting and coordinating CWD and other wildlife disease surveillance programs in Kansas. So he brings a wealth of information to us today, talking specifically about CWD. So with that, Shane, welcome, and I'll turn it over to you.
All right, thanks, Greg. Good afternoon, everybody. We're going to be talking about chronic wasting disease today. I'm going to try to buzz through this as fast as I can so it leaves some room for questions at the end. CWD is just one of my priorities in my job, and I'm a coordinator, so I have field staff statewide collecting samples. Even the public sometimes collects samples for me. Rabies is an important part of my job too. It ties in with human health. Being a fatal disease, we make sure that none of that slips by the radar. And then any sick wildlife in the state is under what I do. We send that into a lab in Georgia, Athens, Georgia, SCWDS, Southeastern Cooperative Wildlife Disease Study, just as a background. Currently, CWD monitoring and surveillance is one of my top projects every year. this year, in addition to that, I'm also having Lance Knoll at KSVDL do the avian influenza testing. We're doing surveillance to find H5 and H7 pathogenic viruses. So we're doing that. And then deer SARS coronavirus 2. If you didn't know, deer get that as well as we do. We're tracking that to make sure that it's not being reassorted and will be given back to us at some point in a different form. Chronic wasting disease. This is a GIF of from 2000 to 2023, designed by Brian Richards at USGS in Madison, Wisconsin. Brian has forgotten more about CWD than I'll ever know, but I'll try to do my best. They put all these years together. I'll let it run twice in case you missed something. The black area in northern Colorado is where we think this originated in 1967. The known distribution of CWD prior to 2000 was in free-ranging wildlife in that dark color as well. So you'll see Colorado and Wyoming prior to 2000. The gray areas are free-ranging populations, geographic spread of CWD over time. And the yellow dots are captive cervid facilities that have been depopulated. All the animals removed, none put in again. The red dots are where captive cervids still remain in infected premises. They could be tied to an infected premises and allowed to remain because they didn't actually find one there, but a lot of these owners own multiple facilities, and one gets contaminated, we assume the other ones might be as well. This is a recent photo from Wallace County, 5th of September, 2024, taken by Kevin Clagg, one of our great biologists out west. The deer was found in a pen with goats. We don't know how the deer got in there. It wasn't acting right, so we euthanized it, and we tested it, and it was positive on both obex and retropharyngeal lymph nodes. And when you get both tissues testing positive, it's a strong positive. The deer is emaciated. That's a clinical symptom. We'll get to some of those symptoms here in a little bit. It's somewhat poetic because we think this may have been similar to what happened at Fort Collins, and I'm not going to point fingers today. A lot of this is my own opinion, but sheep and deer mixed together with something you didn't know about possibly mutated into what we know as CWD. Scrapie may have mutated into CWD, but we don't know that and probably won't ever know that. We get a lot of pictures about sick deer. They're not all CWD. If we get a picture like this out west, probably going to be highly likely it's CWD, but I'll get a report of an emaciated deer in Johnson County, and they'll immediately jump to, oh, I have a CWD deer. Can you come take care of it? Usually it's caused by something else. Hemorrhagic disease destroying the rumen lining will result in emaciation. That's what we've seen out east. This is probably the strongest, this is my statement, strongest one of the day. CWD, if it isn't already, will be the worst wildlife disease America has ever seen. And when I say we, I always mean scientists. We now know it's not going to come and go like viral and bacterial driven diseases. EHD comes and goes. CWD will never go away because it contaminates the environment with prions, and it just continually, deer give it to deer. It has a long incubation time, and it's not going to go away. I actually compare CWD to glaciers. They move slowly across, grinding away at populations, whereas hemorrhagic disease is like an avalanche. It comes and goes, destruction, then it's gone. CWD is like a glacier. This is probably the busiest slide I'll show you today. I'm going to read this, but this is what makes CWD, or some of the things, there's more, what makes CWD the worst wildlife disease that we've seen. It's caused by the tiniest of things, 15 nanometers, smaller than most viruses. Number 2 there is disease prions convert normal prion to the disease form, and that's the problem. It accumulates, it can't be broken down, it causes neuronal death. The prions incubate in the host animal without outward symptoms, so you don't even know you have it for 2 years, and then it kills the animal after 2 years without immune response. The animal does not react to this disease like it does with viruses and bacteria. Prions are excreted by the infected animals. They bind to clay. They become several hundred times more infectious when they do that. They're in the soil for 3 years, that we know as a science, but it's probably forever. And some people hesitate to say that because that's still going on, and I'm sure that research over time will show that. But prions are taken up by plants. There's another problem. That's deer forage, that's hay and commodities too, so keep that in mind when you think about human risk down the road. Prions pass through scavenger digestive systems. They're dumped in new places. You don't even need a deer to go somewhere else. You have that. Prion strains, very important. And the person that knows about prion strains is Dr. Glenn Telling. He worked under Stanley Prusiner, and Stanley Prusiner got the Nobel Prize for finding out about prions and how they kill things. So when you have a strain of something, that can result in a zoonotic threat. And we haven't seen it yet, but we're always keeping our eyes open. And CDC is watching this very closely. The prion is very stable. It resists breakdown. In the environment, you're not going to have 1,000 degrees Fahrenheit, and you're not going to have 40% bleach over the landscape. So it's going to remain there indefinitely. CWD has 2 forms of disease characteristics, frequency and density. A lot of diseases are just density dependent. More animals, more disease. You go in a room, you have flu. If you have a lot of people in there, you have a better chance of getting it. Whereas the frequency depends on contact rates of the deer licking each other. Males and bucks and does getting together, bucks licking each other before they fight, contact rates. The older a buck gets, the less it makes contact with other deer. They just behave differently, and we found that if a deer lives to 6 years old, chances of it getting CWD are very nil. It's not as probable. The dependent disease character, we could talk about that for a long time. There's a lot of things about that. But anyway, it's a hybrid. CWD always leads to the death of the animal. A lot of people say, well, I never saw a deer die of CWD. Well, you won't see it die actually of CWD, but predators take them out, and they'll get aspiration pneumonia from excessive salivation. They're breathing that fluid into their lungs, and they'll get infected and die. And they get emaciated and weak, and they die of exposure. I'll show you a picture of that, and organ failure. Predators and dehydration have seen it over and over. Predators take them out. A lot of times before we'll detect those. Here's a good one, number 11. After a few years of low prevalence in population, prevalence increases exponentially. You can have this disease for years at a low prevalence, and you just don't think anything happening. Then all of a sudden, boom, it's like a spike. The human population is a good example of exponential growth. We remained pretty stable for hundreds of years, and all of a sudden the curve went up. That's exponential. We don't know what that exponent is, but that's what it is. And we've had 50% in the Northwest in older bucks, and I'll show you that in a minute. And 80%'s been documented in Saskatchewan. 8 out of 10 deer in the area with it. Of course, they're not going to die until 2 years, so they have time to reproduce, so you don't really notice the population change until several years have passed by. Natural spread in the population, it goes undetected. It usually becomes too late to eradicate it once it's been there so long. And a lot of diseases are like that. A lot of people tell you get a disease in wildlife, it's a no-win deal. You're not going to get rid of it in most cases. There would be exceptions, maybe with foot and mouth disease. If it got in there, I think we were able to eradicate that in California in the 1920s. So that's rare though. Most diseases, once they're there forever. Natural spread within a population is low, and it goes, okay, I covered that. And 11 is basically the same thing, but the first states to be affected had no baseline of understanding, didn't take it serious enough, so now you can't control it. It's out of the box. Number 14, CWD is known to decline the cervid populations over the long term. We'll get to that. 15 is a social aspect. You go online, you'll find all kinds of information about it. It's not a threat, it is a threat. It's bombardment. It pits believers against non-believers, hunter against hunter, feed supply companies against agencies. All the stuff you probably need to be doing we're all fighting about it while the disease continues to spread and prevalence increases. And lastly, there's no cure for it. The most brilliant people on earth have been working on this for decades, and we haven't found a cure for it yet. They think if we ever do find a cure for it, we're also going to cure Alzheimer's and all these other dementia diseases in humans. They'll all be kind of related. So now we're going to get back into this. TSC is a transmissible spongiform encephalopathy. Examples would be BSE in cattle, scrapie in sheep, mink, have their form. Humans is CJD, cervids, of course, CWD we're talking about, and then there's other ones. What you have are prions converting normal prion to the bad, and they accumulate, causing neuronal death. And the brain becomes a sponge, basically, because you have all these holes in the tissue, and that's where it gets its name. This is a quote from Dr. Elizabeth Williams in 1996 after CWD was discovered on a game farm in Saskatchewan and asked what they should do if it spills over to wildlife. Well, she says, you'll have to be aggressive, remove all sources, all potential movement, cut wider and deeper than you ever think necessary. The deer will come back, but you'll get one chance. If it gets established, you're going to have it for a very long time, and she's exactly right. And this is before we had that map that I showed you at the beginning where it was all over the place. What does she mean by cut wider and deeper? She means kill everything there in that area. Kill all the deer, remove them all, and you might have a chance of getting the prions out of there before they have a chance to bind to clay and pass to other animals that are taking it other places, like dispersing yearling bucks. Unfortunately, and very sadly, Beth and her husband died in a tragic vehicle accident in December of 2004, 20 years ago. We lost a great ally in the fight of CWD, and I often wonder where we would be today if she was still with us. In 1978, Dr. Williams was the first to classify CWD as the TSC. It was a few years before Stanley Prusiner won the Nobel Prize and found that the infection agent was indeed a prion. And Dr. Glenn Telling worked under Stanley Prusiner, and he knows a lot about the strains, as I said. Some have believed the disease agent was not a prion, and you'll hear this sometimes. It's kind of dying away, but there was a guy from LSU, I think his name is Dr. Bastian, and I'm not criticizing anybody. If anybody comes up with the answer, more power to him, but I think he was thinking that this was a spirochete bacteria that was cloaking itself with proteins. And to my knowledge, that's been proven wrong, but there could also always be something that comes up new in science. We always find stuff, so we'll see what happens there. But right now, it is a prion. 99% of people believe that's what causes CWD and these other TSCs. Quickly, the origins, 1967 in Fort Collins, probably mule deer, scrapie mutation. That's up for debate, but that's what we think right now, most of us. 81, Colorado finds it first in the wild elk. Then 85, Wyoming finds it in mule deer. And then 86 was elk in Wyoming the next year. In year 2000, I was actually working for Nebraska Game and Parks, and I was on that hunt to or coal to go get some samples from deer in the panhandle, and we found one with it at 104, I think is what we sampled. So we knew it was in Kimball in 2000 area in 2000. 2001 was Kansas's first detection in a captive elk in Harper County. The herd was depopulated, and we never found CWD outside the wire for years. And so I consider that a success right there. You got in there, we did what Beth Williams was telling us, go in and cut wider and deeper and got rid of that herd. And in 2005, we found our first wild deer with it. It was a doe in Cheyenne County. 2.5-year-old doe. Probably came in from Colorado or Nebraska in natural dispersal. That's what the prion looks like. This weird thing that's made up amino acids, folds on itself. It's a protein. We need them. We have healthy prions in our body. Everything does. And they evolved to keep nerve cells well insulated. And to take that a step further, I read an article a long time ago that it said that they evolved to manage pain, they think. It helps the body react to pain. And so through natural selection, if you know you're hurt, maybe you could do something about it or not do something that hurts you. And those go on to survive and reproduce and so forth. But you do need prions. It's just when they turn bad is when things happen. You get the PRP-C on the left, if it's your left screen. That's normal cellular prion protein. And the PRP-SC on the right is SC is for scrapie, but it just means abnormal prion, bad protein. So it ends up there, and then it converts the normal cellular protein to the bad form. It's not too hard to understand. If you dive into the dynamics of these prions, that's when it gets complicated, and we're not going to go there. But it accumulates in the CNS and causes neuronal death. And you end up with animals that are excessive salivating, classical sign. This is from Wyoming, emaciated white stance, lowered head, droopy ears, and dripping from the mouth. As far as our state goes, I've done this long enough to know what to look for. And I tell our staff, I'm getting more and more reports of this happening if the deer actually has CWD. And we kind of go off of this when we find a deer and the landowner wants us to put it down. The county where it's observed, west or east. If it's Johnson County thin deer, like I said, it's probably not CWD, it's something else. But if I get something like that out west, that's an indicator. That's going to fall into the model to predict that animal is CWD positive. Emaciation is always going to be there. Excessive drooling in the late stage. These are late stage symptoms. Last few months of the 2-year disease cycle. Rough coat, very little fat on the organs. The teeth are gone or ground down. They just stand and grind. Blank stare, there's no one home. There's a drooping head. And one that you don't hear much about, and I've seen it quite a bit, is dried velvet on the antlers when the velvet shouldn't be there. It should have been rubbed off a month or two ago. And close to farmstead buildings, close to farmstead structures, corrals and fences, or near water. Those last 3 there, I think maybe are related to avoiding predation. The deer might have enough cognitive ability to know they're in trouble and they don't want to be eaten alive by coyotes and bobcats. I think they're going to these places as a last-ditch effort to survive. So I'm going to show you some pictures of Kansas CWD positives. These symptoms manifest during the last few months of an 18 to 24-month incubation period. So most of the time the deer is CWD positive, you won't be able to tell by looking at it, but these are the clinicals. This was taken by Mike Hopper, a game warden out west. He's still there, he takes good photos. This photo's been used in a lot of different magazines and documents, and books, published. Excessive salivation, Perfect. You can see it dripping off there. Emaciation. See the dried velvet on the antlers? Blank stare next to farm buildings. It's taken by our biologist in Garden City, Curtis Meyer, Finney County. I didn't zoom in on this one, but you can see the excessive salivation. The emaciation doesn't look as bad, but this is January and deer still has its winter coat, covers up a lot of that. But you'll also notice that in the left shoulder of the deer, the scapula, there's a broadhead stuck in there. So the hunter, I'm guessing, got close to this deer because it's clinical, wasn't really fleeing away, maybe got close to it for a shot, hit it high, hit it in there, and then it ran off and the hunter didn't find it. It's one of my favorite photographs, actually. It's in the corral and has all that stuff going on. Abby McGuire's our biologist with Kevin Clegg out west, and this another deer, emaciated, classical. Photo again by Mike Hopper. This is a deer in Rawlins County that just was walking along. The elements got to it, exposure, and it collapses and freezes in the northwest February weather, northwest Kansas. So CWD positive, dies of exposure. Jason Wagner, he's now our biologist at Cheyenne Bottoms. He took this in Ellis County when he was working there, emaciated deer. Our game warden in Decatur County, Daniel Howard, took this, corrals again. And if you look close, you can see dried velvet on that antler if you zoomed in on it. Ken Hensley, he's our public lands guy at I forgot the name of the lake. Out in Trigo County, Cedar Bluff, sorry. Emaciated, velvet on the antlers again, blank stare laying down. Another one by Jason Wagner, close to water, emaciated, CWD positives. Rawlins County is bad right now. It has been for years. Right in the town of Atwood. We'll have a lot of positives reported every year. Going to structures, bird bath, trees there, laying down, head down, not so much drooling. Michael Hopper took this one of a whitetail. This is probably a hybrid mule deer, whitetail hybrid, but it got its antler locked into the fence. It also had a brain abscess, so it was CWD positive, but we don't use this as the poster child for CWD because brain abscesses do this to deer as well. This is pretty gruesome. Deer lose their ability to think right, and so they wander into stuff. This one, CWD positive, wanders into a fence and twists around and chokes itself to death on the wire. And you can see the dried saliva around the mouth. Classic symptom. Photographed by Wes Sowards. He's our assistant wildlife director now. And of course, we're going to get some hunters harvesting some clinicals. Not all the time. Most hunters won't shoot sick-looking deer. They won't want to waste their tag on that. But this one was dry velvet on the antlers again, emaciation. And this photo was taken by a friend of mine. He helps me collect samples in Oberlin, Decatur area. His friend actually shot this deer, and his friend said, "Hey, Patrick, come over and take a look at this. Is this normal?" No, it's not. You shouldn't see light penetrating the body cavity like that, and you can see the ribs showing. So this was a late stage CWD. So most deer infected with CWD appear healthy without outward symptoms during the 18-24 months incubation time. The deer infected with CWD pirons only exhibit those symptoms in the last few months, so I can't stress that enough. Most of our deer that have it look apparently healthy, but more and more out west we're getting high prevalence and more of them are being detected now, late stage, right before they die. So here are some apparently healthy deer, hunter harvested CWD positives. This one doesn't look all that healthy because something's been eating on it, but it has good muscle mass, thick neck, nothing wrong with the antlers. Sherman County. And here are a montage of collection of 4 photos, apparently healthy trophy bucks, Kansas, Rawlins County, and the one in Decatur, another one in Decatur, and then Graham. And then this one, I would say, man, that's beautiful. You know, nothing wrong with that one. No, CWD positive. Beautiful buck, palmation on the antlers. And a former game warden took this one. Looks like it's healthy to me. Nope, CWD positive. Here's one that the body looked great, good condition, but that left eye, but that's not CWD. That's from fighting with another buck and getting poked in the eye. So we'll see that quite often. And a lot of times that's how deer get brain abscesses, from the pokes and the bacteria. Truparella pyogenes getting into the calvarium. This was a young hunter's first deer, unfortunately was positive. This is what we are concerned with, hunters harvesting positives and quitting the sport, because we need hunters more than ever now to keep densities in check. So we'll move on to surveillance and monitoring. To do any surveillance and monitoring, you're going to need some samples. That's a truckload of deer heads. We used to have them all delivered to Emporia, and we would process everything there. This was at the end of doing that. Now we have taxidermists and processor collect our deer for us. They'll collect the sample. We pay them $12 a deer to get those lymph nodes or that Obex or both and put it in formalin for us. We target the older 2.5 bucks. Usually no fawns. It usually takes 4 months for prions to accumulate enough to be detected. And here's where you'd find that sample we need for CWD testing. As most of you know this, the Obex is about 2 to 3 inches in from the entrance of the foramen magnum there, and it's dorsally located on the brainstem. And then the retropharyngeal lymph nodes right in front of the occipital condyles. Another picture of where the Obex is located as it creates the central canal of the spinal cord, that V-shaped form right there. It's where the prions accumulate. If we collect samples from an elk, we'll pull both, because sometimes they will show up in the Obex before they'll show up in the lymph nodes. And with deer, it's kind of the opposite. So we do both tissues for elk and just usually lymph nodes for deer. But if a hunter wants the KSVDL, helps us out, they will test both of those tissues for the same price for us, and they can run that right through, and it gives you extra confidence in the result that way. So we use IHC. We put those samples right into 10% formalin. This is the gold standard. We used to use ELISA here at the lab, but we don't do that because the biorad kits were hard to get a hold of, and hunters kept waiting for results, and we just couldn't have hunters wait months to get a result. They have the deer in the freezer, they want to eat it, but they don't want to eat it without a result. A lot of them are like that. With ELISA, the benefit is you can do several deer at one time. IHC is a deer by deer deal, but it's the gold standard. Like I said, you slice through the tissue, you stain it, or you slice through it with a microtome is what it's called, then you stain it, and then the pathologist analyzes the slides. I'd like to extend my thanks to Jennifer and Tony in the TSE lab and their helpers. They really help us out getting those things processed fast, and they're resulted out. And thanks to the pathologists that read that. Many of the pathologists read it, not just one. The red is, around that is the prion. This one's highly infected. I asked for slides one time and I looked at them. You could hold them up, or I had a picture of one. You could see it. You could see the prions just by holding it up to the light. You didn't need a microscope. a lot of those. So we're going to go into the sampling that we've done over the years. It's sort of busy, but 28 years of sampling, about 36,000 samples, 1,075 positives, and most of those positives within the last 10 years. We really weren't finding it that much early on. You'll notice there in 1996 and 2002, very little sampling going on. Those are sick animals. And we thought maybe I didn't start until 2009, so this is all before me, but they wanted to test those because they're getting this bad information that something was happening out west. See, Colorado, Wyoming had this going on, and we thought, well, maybe we should start looking for it then. So we did and didn't find any. Then all of a sudden, Vet Services wants to fund some surveillance, and because they want to know how much of this is out there, because at this time we didn't really know if it could be passed to cattle. And so Vet Services give us some money for 9 years in a row to do surveillance, and we start finding it towards the end of those 9 years, 10, 15, 10, 8 positives out of about 2,000 samples. So that funding eventually goes away, and then there in 2012 we go back to a surveillance like this. I designed 5 surveillance zones, and so I started in the north central zone there in 2012, and then I think 2 years in a row we had money from 2 different places, so I was able to do 2 zones a year there in 14 and 15. But you can see, the point is, you can see we're finding more CWD with 1/3 the sample size. So you know prevalence is increasing. It's not rocket science. And then 2020, we had a study with MU going on, spatial analysis. Ram Raghavan was using some Bayesian modeling to determine CWD prevalence, characteristics, and spread and direction and different things and environmental variables. So we were able to do statewide surveillance there for 3 years in a row before we had to ended the project and went back last year to just 1 zone a year. But we added a little twist. We are able to offer statewide testing now to hunters as long as the money's there. We have a $50,000 grant with Kansas State to test about 1,500 samples. That's what that means. So I'm going to do the Northwest zone this year, targeted. Any deer I can get my hands on, vehicle, kills, sick, suspect, hunter, everything. And it's hard to get a good sample size there, just not that many deer there now. And then the rest of the state, interested hunters can have their deer tested. They just have to fill out a data card. There's one in our regulation summary booklet. You can get it at Walmart, or they can bring the sample here and fill out a data card here. Client care has been very helpful here. I work well with those folks, and they help me out getting those hunters lined up and what they need to do, so it's billed to us and not to them. So after all that sampling, we want to know, okay, what is it? How bad is it? And the latest estimate for these are 2.5 and older, apparently healthy. So I take out the sick and the vehicle kills. I take them out and just do the apparently healthy hunter harvested male deer, both mule deer and whitetail together. The prevalence with 95% confidence is going to fall in the range of 24 to 45% in the Northwest. And there's some areas in the Northwest that are contributing more to that. So Rawlins County, Sheridan, there might be prevalence of 70% to 80% there now in those areas, whereas the zone as a whole is in that range. And then North Central is getting just about as bad, 25-38. As it spreads east and south, it's actually getting worse than the Southwest, we think. And still below 5%. Keep 5% in mind in the South Central and East. When it's below 5, you can maybe manage for it a little bit better. So once again, those are male deer, 2.5 and older, apparently healthy. This will give you a different view of the clustering of CWD. We've had it the longest in the Northwest, so you would expect the prevalence and the darker shades to be up there, getting darker in the Southwest, North Central. But these, if you can see that cursor, can they see that cursor? No. Okay. The sparks is what I call them in the east. There's one positive in those counties east. That's the start of a problem that we'll see manifest into what we see in the Northwest right now. It's going to take about 17 to 20 years, and the east will look like the Northwest does today. That's my prediction based on what I've seen. But there are some things that might help control that. Another map we share with the public. It's detected in 66 counties now. We have 105 in the state. Prions are likely deposited in every county now. So even those counties that don't have a positive yet, we think it's probably there just below detectable levels, and we don't get many samples, especially the ones out west. I mean, it's a no-brainer. There's probably CWD there, but we just don't get many samples from those counties. So our current monitoring program for CWD is 2 prongs. And I already talked about this. We're going to do the Northwest zone this year, and I'm going to collect whatever I can get up there. And then the statewide voluntary is for interested hunters to have their deer tested until the money runs out. We do test all 6 suspect deer that are sampled by our field staffs on a statewide basis. Doesn't matter what we're doing, we always test those. That zone surveillance prong is to help us determine prevalence trend change over 5 years. Kind of a tongue twister there, but over 5 years, how is it changing? Is it going up or down? Because we haven't been here before. Maybe CWD will burn itself out. I doubt it, not based on what I've seen, but you never know without testing deer and tracking it. So we track it. And the hunters are interested. They're interested in knowing. A lot of them are, about what's happening out there. What can be done about CWD in the next 15 minutes? Nothing can eradicate it. There's no vaccine or other treatment available. Research continues by the best and brightest. We have education programs in place, like in the written narrative of our hunting regulations. A lot of states have pamphlets about CWD, so we try to educate people, and we try to educate them to report those sick animals. Maybe we can remove them from the environment. We reduce some congregation of animals will help, so we educate them that way. We want to bury carcasses if we can, get the prions below ground, away from other deer and scavengers. Landfilling them, carcasses is helpful because no deer is going to go inside a fenced landfill. Most of them won't. Leave the carcasses where you find them. You already know it's there, so don't take it to a new area and start CWD in a new area. Get all the depredation permit deer taken. We have a lot of crop damage in Kansas, so we test some of those deer, and 3 of them had CWD in 2009. Here's an important tool that can be used if you can get support, and that's sharpshooting. Excuse me. The goal is to keep prevalence at 5% or less. Illinois has been doing it for years, probably decades. They have support, but it wears a person down. And Missouri's in the kind of early stages of what Illinois has gone through, and they seem to be having success there. Wisconsin started sharpshooting, but they ran into a political juggernaut and had to stop, and then their prevalence increased after that. New York went in and shot some deer when they first detected it, and they think they eradicated it. They're the only state in the nation who thinks they eradicated CWD because they took Beth Williams' advice, went in and got rid of them in that area around the captive cervid farm. And they haven't detected it since there. If you don't have public support, and most states don't, mainly because the disease can't be eradicated, you're not going to convince them to start sharpshooting. And you need to realize once you have sharpshooting in place, you're going to have to do it forever because it's not going to go away because the environment's contaminated. You're going to be doing it for a long time. I've talked to people who were on sharpshooter teams in these other states, and they'll flat out tell you it causes staff fatigue, family stress, divorce, and they'll say it isn't hunting. You know, we all think of going out in the ground blind and hunting deer. When you're sharpshooting all the time, it just doesn't, it's not hunting anymore. It's not fun. And then, so we try to keep promoting deer hunting to keep densities in check. That's why we need those hunters. We need hunters to control deer, take antlerless deer. There may be a time, and I think we're going to start cutting back on the antlerless permits in the Northwest and West because we're losing some population out there, not just because of CWD, but because of habitat. If we don't dial back the doe harvest out there, we could see possibly localized extinction in areas. And Wyoming, there's places in Wyoming that are saying that there won't be any deer left in 20 years at the rate they're seeing them go. But right now, nationwide, it doesn't seem that extinction's going to happen. There is some resistance out there. And then for us, hemorrhagic disease, which is going on right now, it's not too bad yet. That's caused by a biting fly. It's a virus. That may actually help us control CWD in the east by taking out deer. If the deer are weak and hanging out by water, the biting midges may transmit virus to them and take them out, so they're not dumping more prion into the environment. So it's rather complex. We haven't seen that yet. Some natural helpers are lichens. We don't have many lichens in Kansas that do much for us, but they have I think an enzyme or chemical that denatures prions, known for a long time. Probably that research was probably done 15 years ago. Bobcats and other cats. We know that prions are denatured in bobcat guts. They do not pass through the bobcat. Other cats, I was thinking maybe mountain lions are similar to that, but I'm not sure on that. That research is probably still going on. And then humic acid. The more plant litter you have, the more humic acid you'll have. None of these will likely change the outcome on a big scale. Some of our research will leave question time. This is a graph of Rom's research identifying some prevalence trends over time using Bayesian modeling. He's also finding some factors related to how CWD spreads and the prevalence, and that's still there. A lot of those factors we just common sense, like close to water, soil type, we knew that. So we would probably have to continue this research to tease out the more important ones. Andrew Hope, right here on Kansas State, geneticist, he's looked at some alleles in our deer herd, and we find resistance, greater than expected resistance. So that's a plus, I guess. The problem with resistance is the deer lives longer. It's still going to die of CWD, and it's dumping out prions on the ground. So if a deer is infected, you really don't want it to live very long. You want it to get gone so it's not contaminating the environment. This is our map from our slide from our deer study we did out west. Two populations in the west, and male deer there, whitetails and mule deer, 38% and 35%. That's what we're finding. That supports what our confidence intervals out there are in the west. This study wasn't just directly searching for things about CWD. CWD was a side thing. They were looking more at habitat, and fawn survivability is not good, and it's habitat related for the most part with stem heights and dryness and those types of things. So it's not all about CWD, but CWD is definitely contributing to population problems out there. Risk to the population, some papers here. It's causing declines in Wyoming. This is 2017. 3 studies have shown population declines. these populations would have been stable or increasing without CWD. So CWD is having an effect. And most states agree that if you get to 30% prevalence, you're going to have a population decline. So what's that mean for Kansas? Well, there it is. There's we operate on a deer management unit when we do deer management things. So 1, 2, and 3 are in the northwest zone, if you look at it that way, right where we have a lot of CWD. Here is deer management 3. You can see the trend is down. And I'm not saying this is CWD, but it's down. CWD is playing a role here. Maybe not the entire picture, but, and then unit 2 headed downward, and unit 1 headed downward. So the gist is the populations are decreasing up there, but we really don't know how much is attributed to CWD. Risk to humans, unless I can run this in 5 minutes. What's the risk? Well, it's similarity to other TSEs. So BSE killed 200 people, and it probably will kill some more in the future as it incubates in a few people. Number 2, laboratory findings are not all promising. 3, you have unknown incubation times. What if the Trojan horse comes out in 20 years? What if it's 10? What if 5? We don't know. So, and then prion strains, where you take prions from one animal, pass them through, and then give them to another animal, then that animal gives it back. You know, it's really, the risk is not zero. That's the best we can say. So if you know your animal's positive, do not eat it. The risk is not zero. There's a cow with BSE. This is a graph from a long time ago. 5 of 13 cattle got it from mule deer prions. 12 of 13 cattle got it from whitetails, and then mule deer right to sheep. The thing to remember here is don't panic and say, oh, they got it. These are intracerebral injections of prions right into the brain. And it's pretty much agreed that if you inject anything into somebody's brain or something's brain, it's going to get sick. And so, but it is. This is another, adds to the risk is not zero. And then this one here was a bombshell about, when was this? 4 or 5 years ago, 6 years ago. Macaque monkeys, that's the closest relative we have to humans that can be worked on. We can't work on chimps, and so we work on macaque monkeys, and Canadian study found that 2 of those macaques died they contracted CWD from eating positive deer meat. They were euthanized. They got to a point where they had to euthanize them. It's not published yet. So to my knowledge, and I had heard, and this is my opinion, I heard that maybe they had underlying conditions. Could have been some diabetes in those macaques, not sure. But until it's published, we don't really, once again, the risk is not zero when you see something like that. Conversely, NIH finds no chronic wasting disease doing the same kind of study. So you have countering studies finding 2 different things, finds none in this study. And then CWD is able to cross the species barrier to convert human brain cellular protein into the infectious form. In vitro, this is mice. These are humanized mice studies. So once again, risk not zero if you're finding this. And then the bright side, the last recent paper in June of this year was they were unable to get prions to transmit to human cerebral organoids, basically models of organs that they design in the lab, and they could not get the prions to transmit to those. So that's great. So that leaves me with this question and answer session. Once again, I'd like to thank the client care desk for all their help in getting our samples, and Jen and Tony and helpers in the TSC lab, Greg and Jamie administering all this, and then the folks in billing helping us get our bills paid on time. And then Lance Knoll, got to throw him in there because he's helping us with avian influenza this year at the lab. So we look forward to more collaboration, and it's a great place to do business. Appreciate it.
Thank you, Shane. A lot of good information. It may just be me, but it was kind of a bummer or downer. Looking at your first map where it goes from Kansas, Nebraska, all the way up into Canada, how in the world are we ever going to stop the spread of this, especially for the fact it can live in the soil for years? And what are we going to do?
We don't know.
Okay.
The Canada place is, they think that got up there in the captive cervid industry. The elk that were taken up there to farms started that problem. So what we can do is try to prevent, knowing that and knowing some of our stuff, try to prevent it from getting into new areas. And other states are finally getting on board. They're passing a lot of regulations to stop it from getting to new areas. Once you have it in an area, I don't think there's anything we can do except watch her go and hope for the best.
So knowing what you do about CWD, do you recommend that all hunters test their deer, even like in eastern Kansas where it looks like the prevalence is extremely low?
Yeah, I try to follow what CDC says. They say if you're hunting in an area where CWD has been detected, you probably should be having your deer tested. We do not want people eating positives, unknown or known. We have folks that don't seem to care and they'll eat one if they know it's positive. That's up to them. That's their choice. We don't recommend eating sick animals.
And it looked like there was some genetic resistance to clinical signs or from time of infection to disease. Is there any hope that there's some deer populations will have resistance to the actual infection?
That'll be natural selection, and then nature will take over and fix that, but we might not have many deer left by the time that happens.
And then your comment about the goal is if you can keep the prevalence below 5%, you can manage it. What does that mean? What do you mean you can manage it?
It just is not going to affect your population growth and size, and you're going to have a lot more. You're going to have 95% of your animals healthy. So the chance of a hunter harvesting a positive is null and void, and you're below that exponential growth curve when you're keeping it at 5. If you let it go higher, then she shoots up and you can't do anything about it then. That's the best way I understand it.
It looks like there's certain areas where the prevalence is so high that the number of hunters may decrease, and if that increases, what is the Kansas Wildlife Parks going to do about this if there's not enough hunters to keep the density down?
I think they're talking about that in meetings now, at national meetings, not just our state, but what will we do in the future if we tend to lose our deer hunters? Because deer hunting runs not only our agency, it runs a good portion of the Kansas economy. People coming here from out of state to hunt, hotel stays, restaurants. There's an economic benefit to having deer hunting around to the state.
How many deer are typically harvested in Kansas every year?
80,000, anywhere from 80 to 90,000. The highest was in the year 2000, I believe, and we harvested 120,000. And still, it's relative. So Pennsylvania has more deer killed on the road vehicle accidents than we harvest in our deer hunting.
Wow.
So there are a lot of other states with a lot more deer.
Is there any work to develop diagnostic testing that can be done either in the environment or surveil large populations of animals that are still alive?
What was that again, Greg?
Yeah, my questions are is there any work on diagnostic tests that can, like
Field
we do,
test?
like pigs, we can use saliva from ropes where they chew on. Is there any environmental or anti-mortem tests being developed?
Well, they have some anti-mortem tests now, but they're not as reliable. They're not as sensitive enough to catch it. So they're using that in captive cervid farms because you're not going to kill your animals. You're going to test them live. But they're looking at things like RT quick analysis where there's an assay that can pull a little bit of soil from the ground and tell you whether there's prions in it. So look, a lot of it's looking at the environment. If it's there, you know it's there. You don't even have to have a dead animal or a sick animal.
Okay.
And then maybe you can manage
Yeah.
differently that way.
Yeah, I think
But
that'd
that's a hard one to answer
I think
because
that'd be important going forward.
well, from a field biologist standpoint, we're waiting for the field test, which won't probably be as good for you guys because we won't have to test as many deer. If hunters have it in hand and can test their blood of their deer and say yes or no within a few minutes, that may be what'll happen in the future. But that isn't there yet. They're using gold nanotechnology in Minnesota, and they think they have it. It's just how are they going to deploy that to the field and not charge a lot for it? Because most hunters aren't going to pay too much for that.
Okay, our time's up. Again, Shane, very much appreciate the information on CWD. Everybody see Shane's email address at the bottom of his name there. Please, he's said he will answer all your emails, so flood him with emails, questions about CWD, and we'll see you at our next webinar. Should be a month after next.